Welcome, everyone. It is the middle of May, and we are very pleased to welcome Dr. Hans Gregerson of the California Medical Innovations Institute to our installment here of Bridging the Body and the Brain Spark seminar. My name is Susan Tappan, and I'm part of the Spark Data and Resource Center. And the purpose of these seminar series is to introduce all of the great research that is enabled and shared on the Spark platform called the Spark Portal. Dr. Gregerson will be speaking about the winding road of commercialization of a Spark-supported device. And it's a very fascinating story. I'm very excited to hear all of the intricate details of it. But before we get started, I'd like to invite you to ask questions using the question and answer panel on the controls that you have in front of you. And if you have any questions that come up during the seminar, just go ahead and ask them. We'll hold them to the end. And at the end of the seminar, I'll be a bit of an MC and guide Dr. Gregerson in the questions that you guys have promoted and posted for us to consider. So, the Bridging the Body and the Brain is a Spark seminar series. Dr. Gregerson is our second seminar. The next one is coming up at the end of the month, and you can see that we have seminars all the way through to the beginning of September. We're very excited about the entire series and just how broadly the topics that we will be talking about span everything that relate to how the central nervous system communicates with the peripheral nervous system to control organ and body functions. And so we find that to be a tremendous area of interest, and the topic has a great wealth of detail. So the Spark platform, the Spark portal, is an open neuroscience and systems physiology platform. It is an NIH approved and HEAL compliant repository. So data that you collect during the course of your experiments can be aggregated and shared fairly on the Spark platform. You can do other things besides just place your data on the Spark portal. You can view other data and models. You can see device information as well as other tools and resources. You can view data in the relationship with body space across multiple species by using the interconnectivity maps that are created on the Spark portal. We also have a computational platform as well. It's called OSpark. It is spectacular, and it can allow you to build and explore modeling and data analysis pipelines. So you can take data that you find on the Spark portal and port it over to the computational platform in order to do analyses. We also encourage you to contribute. So as a part of the community that's interested in systems physiology and PNS connectivity, we accept data, devices, and models about the peripheral nervous system, and we are compliant with the 2023 NIH data sharing mandate. Although I've mentioned that we are NIH compliant, NIH approved, and we're accessible for the NIH data sharing mandate, we accept data from across the world. So we have no restrictions on where you are located in order to contribute your data. If you think your data has a good fit in this company, we would like to encourage you to contribute it. Additionally, being able to make the data harmonized across the platform, regardless of what species or scale you're studying, ensures that we're building a nice, tight-knit community of researchers who can explore areas that they're interested in and find new things to stimulate new questions. So if you would like to contribute data, we accept individual submissions. They undergo professional curation. We reserve a DOI for you, and it allows you to build your body of work. It can include code and simulations. One of the nice things about a data set that is contributed to Spark is that all the data comes and stays together. With this repository allows us, by the way that we've created it, to be multi-scalar, multi-species, and multimodal. So if you've got imaging and physiology data, you do not need to break them up into two different repositories. All of it can be controlled together and accessed at the same time from Spark. We also can help you with grant language and fulfilling the DMS policy guidelines for NIH applications or other applications to different agencies that require a fair data sharing component. We're very well versed in that, and the professional curation that we provide ensures that your data gets the highlight that it deserves. So far, we have over 11,000 samples across 4,000 subjects, spanning 26 methods, 54 tissues, and 7 species. These are from over 600 unique contributors, and we're hoping that you will decide to become one as well. All the data is presented together, and we ensure that you can hop between different areas of interest by using our tools on the portal to make search and location straightforward and facile. We have 237 research data sets, 46 anatomical models, and 46 computational models. All of these are 100% available for reuse. You can find a data set that you're interested in, utilize it in your own research, and contribute your novel findings back to the Spark portal to continue to push the boundaries of what we understand. The data sets that are contributed to Spark are heavily accessed too. We have over 12,000 downloads of data sets and models to date. Research data sets are very popular, but the anatomical models and computational models are also heavily accessed as well. Across the x-axis, you can see the data sets are presented across the x-axis, excuse me, and that there are a number of different types of data sets that are downloaded, and even recently contributed data sets, those that are furthest along the x-axis, can be quite popular. So as I mentioned, we're going to be hearing from Dr. Hans Gregerson today, and we're quite excited to hear his perspective on the winding road of commercialization of a Spark-supported device. Hans, I'm going to give it to you.
Thank you. Let me just share my screen here. All right, it seems it works. Thank you, Sue, for the introduction. It's a pleasure for me to give this speech. I've been part of the Spark program from the early days when it started. I'm still part of it, but I don't have current funding. But we do upload data to the Spark portal. I think we have uploaded 6 data sets and 2 models. So the agenda for my talk, these are just some broad terms, but I will provide a background, my own background, also physiology and medical background. We will discuss the innovation process where the Spark support came in. And then I would suggest, if you're not that interested in anorectal properties, that you hang in until the last 5-6 slides, which are more on the FDA process, the challenges we met, and the clinical adoption. So my background, I'm an MD PhD from Aarhus University in Denmark. However, right from the beginning, already as a student, I started on medical research. I was more interested in bioengineering, and bioengineering is really a merger of all kinds of engineering, material science, mechanical engineering, electronic engineering. And I kind of saw a space between medicine and engineering where I could contribute. I also took an MBM degree, a Master of Public Management, because I was interested in innovation and in leadership. And I worked at universities in different countries, as you can read here. Since 2016, I've kind of been working primarily in United States. I've been professor at California Medical Innovations Institute since 2020 and became the president or CEO of the institute a couple of years later. But my life has really been about innovation and about one particular impedance technology. So I was introduced to a technology in urology called the field gradient principle. So it was a simple balloon distension technology where you could measure the pressure and cross-sectional area or diameter inside a balloon you would expand in the urinary tract. I grabbed that technology, translated it into a larger device that could be used in gastroenterology, which is my field. It was not something that could be commercialized or patented, but there were certainly products that could come out of that. So in 2001, we invented what we called a multimodal probe. So it was like a test of different pain mechanisms in the esophagus. And that technology was acquired by the Californian company Alpine Biomed in 2008. It was never commercialized, even though it was acquired, because Alpine Biomed got acquired by Sierra, another company, and they had a different strategy, so they kind of laid it off. But luckily, they gave everything back to me, so which they were not obligated to do. A couple of years later, I invented also an impedance technology. We call it the functional luminal imaging probe, or FLIP. So it was for studying the lower esophageal sphincter in relation to reflux from the stomach into the esophagus of acid. That technology was acquired by an Irish company in 2007. They got the FDA approval, the CE mark, and commercialized it. But it's still, I think they had the FDA approval in 2010. It still took 7 years before Medtronic acquired it in 2017, after several due diligences and so on. And I'll come back to why that took so long time. What we're going to talk about today is the Fecobionics device. So I'll come back to that, and also about how we develop new applications and products from that device. So first, about the physiological and clinical background. We all go to the toilet every day or every second day to defecate. So what we eat, I mean, some of it has to come out again. And for the majority of us, it's a simple process. We're used to it, we go there, it takes a few minutes or longer time. But it's based on a quite complex neuromuscular coordination. And there's a great deal of the population, you can see numbers, 10%, 20%, 30%, who suffer from incoordination. So they may have weak anal sphincters or be hypersensitive, they develop fecal incontinence, or chronic constipation or dyssynergia of the pelvic floor. And the incidence is increasing because we get older and older. And for example, half of the people in elderly home suffer from fecal incontinence. So it's quite common. The good thing is that these disorders or symptoms can be treated, for example, with biofeedback therapy, where you retrain the muscles in the pelvic floor. If we look at this more from a market perspective, there's about 260 million adult Americans, and about, let's just say, roughly 25% of those would suffer from these symptoms, so again, chronic constipation, fecal incontinence. But not everybody needs treatment, and this is underdiagnosed because people are embarrassed going to the doctor or even visiting their friends if they suffer from incontinence. But we believe that about 7 to 10% need therapy one way or another. Therapy, of course, requires a proper diagnosis. And the way we see the market, can we just capture 5%? That is potentially 1 million patients every year who need diagnostic procedures. And some of them will need biofeedback therapy. So there's more than $600 million market potential alone in United States. So let's talk about what we wanted to develop here or change. Well, first, from a scientific view, I wanted to get a better mechanistic understanding of the defecatory process. I didn't believe that the state-of-the-art technologies on the market would give you good data. And there's been a lot of studies, of course, that support these technologies, but also speak against the technologies. And one thing that has been found is that there's considerable disagreement between the results of these tests, and they correlate very poorly with symptoms and treatment outcomes. So we believe there was an unmet need for an integrated test. So if you could do all these tests just in one session, that would, of course, reduce variation between tests. It would also save cost and time. So first, what is it we want to measure in anorectal physiology? Well, defecation is a mechanical or mechanosensory process. So since it's mechanical, you want to measure what is the driving force, how much do you push to get the feces out of your body when you kind of have the urge sensation. But there's also, so that will come from up here from the abdominal muscles, but there would be resistive forces here from the anal sphincter. And there is a puborectalis muscle that kind of give the anorectal region an angle. So that's called the anorectal angle, which is also important to measure. The more acute it is, the more difficult to pass feces through the region. So we wanted to measure force, anorectal angle, displacement, shape of the rectum and anal canal, rectal sensitivity, and the evacuatory flow. And as I mentioned, we wanted to develop a device, a single device that could measure these things. So let's look at what is the state of the art on the market. You see that one here in the upper left corner, that's called high resolution anorectal manometry. It's a catheter that's inserted into anorectum. It measures 12 to 24 pressures along the catheter, and you usually display it as these color topographies that most gastroenterologists now are used to. So that is the state of the art, maybe currently the most important test for anorectal or defecatory function or dysfunction. But I call it a static test because it's based on a catheter. And the same with this flip or EndoFlip device. That was the one I developed that is now with Medtronic. It is not a state-of-the-art test, but some laboratories do it. They get one pressure, but they get a diameter profile when the patient do various maneuvers, for example, squeeze the anal sphincter muscles. Then we have what I call the expulsion test, and one is the fecography, which is a radiological test. It can be done with fluoroscopy or MRI, and it's used for measuring the diameters of the anal canal, of the rectum, but more importantly, this anorectal angle we talked about before. The downside is it is radiological, it requires imaging, radiation, and you have to interpret the data. Then the last test is called a balloon expulsion test. It's an extremely simple test. It's a small balloon you insert in rectum. You fill in 50 milliliter. If the patient can evacuate it within 2 minutes, you're normal. If it's take more than 2 minutes, you're most likely constipated. So these are the 4 main tests of anorectal function. And this is kind of where we wanted to integrate elements of each of these tests into this Fecobionics prototype that I'm going to talk about. First, a little bit about the innovation cycle. This is a diagram I downloaded yesterday from the internet. You can find hundreds of different variations of these diagrams, but it's kind of takes us through the process of getting the idea, put it in context, build prototypes, protect the IP, go to the next level of prototypes, final product, take it through regulatory, and then you get into the whole commercialization process with marketing and distribution, reimbursement, which I will touch upon later too. And it is a long process. So I first got the idea for this Fecobionics device in 2009. So it's 16 years ago. And you may ask, why didn't you move further on this? Because I really thought this was the best idea I ever had. Well, I went into a new position as dean of a university. It was totally administration leadership for 3 or 4 years. So it took like until 2014 before I could move on. But first about what I thought about in 2009, a device that would look like feces, that would be bendable like feces, that would have pressure sensors at the 2 ends. So we would measure pressure in the direction of the flow. All other technologies nowadays, they measure radial pressures on the side of the catheter. But this is an insertable device that would measure pressure in this direction, which we found more relevant. Furthermore, it would have motion processor units, so it would have gyroscopes and accelerometers at both ends, and from the directions, from the orientation of these MPUs, we could compute how much does the device bend. And if you know the bending of the device and it passes through the anorectal junction, then you would get an electronic measure without the need for any interpretation, just from these data. So no need for radiology. So, but as I mentioned, it took 5 years. I moved to another position and I started myself to build some prototypes. So I would buy some silicone resin, play with the shape, the length, the diameter, and think about how much electronics I could put into this device. And I went through that stage in 2014. I was convinced here is something. So we filed the first patent application. Now we have several patent families built around it to give us even better protection, and some ideas came up later. But I'm not an engineer. I am basically an MD. So even though I know quite a lot of engineering, I am not the one who design and build devices. So what did I do? I contacted a collaborator at a Danish university. I said, do you have some students you could send to my current laboratory, which was in China at that time. So within a few months, I got some young students, to hardware, to software, to in software. And they worked with my Chinese team to build this device we see here. So that's what we call the wired Fecobionics device. So it would have the pressure sensors here, as I mentioned before, the motion processor units, some extra boards here. And we would go directly from bench studies to humans because we consider this to be a non-significant risk device. Because normally you would first try it in large animals, is there any safety issue and so on, but we felt quite safe about this. We got it through IRB approval several places and could do the first studies in humans. So the IRB committees agreed with us, this is really non-significant risk. We did the first studies in Denmark and in Hong Kong. So this device was 10 centimeter long, 12 millimeter diameter, and here you can see how small some of these electronic components are. And this is just like a short video how it would work. The device would be inserted into rectum. It would be wireless at some point. The first were wired, as I mentioned. We would distend the bag until the patient would feel the urge to defecate, where they normally would go to the restroom. And then the device would transmit data to a computer. And after we leave the room, it would be evacuated and we would have measured all the angle data, the pressures, the shape, and so on. And this is kind of the point where Spark got in. I spent more time in US. And I got aware of the Spark program, and they had, I don't remember exactly if it was an OT1 or OT2, but they had a call on technologies to understand the control of organ function by the peripheral nervous system. And we got an award in 2017 for Fecobionics device for mapping colonic and anorectal neuromuscular function. I saw the clinical applications in anorectal, maybe also some in colonic, but I would say the Spark program was more interested in colon, maybe because it has been less studied. And we did colon studies in animal experiments at that point, and we did a lot of studies on humans on the anorectal application. And lucky for us, there was another call the year after, where the PI would partner typically with a medical device company to develop new clinical endpoints. So something that should go beyond what is kind of the primary endpoints. So we got a grant where we studied neurophysiological mechanisms in sacral neuromodulation. Sacral neuromodulation, or SNS as it's called, is indicated for patients with severe fecal incontinence. There are several device manufacturers. One is Medtronic, and Medtronic was our sponsor. It's an implantable device. It sends out electrical currents, and that apparently stimulates either the anal muscle or rectal sensitivity. But there was a lot of debate about the mechanisms, and our study looked into that. So what came out of this grant was the device you see here at the bottom. And it was kind of totally re-engineered from what you saw up here. One thing, it looks a lot better, but that's not the most important. I mean, it was smaller, it was built according to standards, it was wireless. The algorithms for computing the bending in 3D space were improved, and so on and so on. Basically, everything was redone. And here you see the device on the left side here. You can see when I kind of move it around in space, how it kind of changes on the monitor. So clearly we can measure the orientation. And on the right side here, yeah, so you can see now I bend it, and you see in this integrated view how it bent downward and upwards, and you see a graph where you get the actual values here. So it seemed that it worked. Again, when you develop a new thing, you have to compare with the current technologies on the market, and I already showed them to you. So we kind of made this table here where we looked at what can we measure with Fecobionics and what does the other technology measure. So we call this features. Fecobionics, the diagnostic version, would measure 14 features, the therapeutic version 11, which was way, way more than any of the other technologies. So we are truly integrating what these other technologies are doing. But so there's some validation required by FDA, and I'll come back to that. This is what I call non-regulatory validation study, because when you develop a new technology, you need to convince people that it measures correctly. And they would also be interested in how does it measure compared to the state of the art. So we published a number of studies where we compared it with that balloon expulsion technology, with anorectal manometry and difficography. And in most cases, we measured kind of the same thing, but not necessarily always, but that could be explained by different procedures. We would study association with symptoms and how does that compare with the state of the art. Some more scientific studies on where we integrated the acceleration signals, got velocity when it was moving and position at a given time point. So that was really related to the accelerometer data, comparing evacuation in upright versus laying position, repeatability, tolerability, safety, which of course is a major concern of FDA. So we did about 100 studies or more in normal subjects where we would look into all of these issues, and we would develop new metrics. The last one, proving the device consistency, I'll come back to that on the next slide. We did multiple clinical trials in patients with fecal incontinence, constipation, low anterior resection syndrome, which is a syndrome most patients have after they got removed low rectal cancer or anal cancer. Currently, we have 3 NIH-funded R1 grants on the technology, where we are studying 260 patients, and we are doing 3 trials abroad. And to cut it very short, the overall conclusion is the device is superior, and in some cases you can say non-inferior if it shows the same to current technology. And that relates to repeatability, how tolerable is the device for the patient. So we ask them to fill out questionnaires. How does it correlate with symptoms reported by the patients? What is the sensitivity of the device, data integration, and so on. All of this is absolutely necessary because people will ask you these questions along the way. So I mentioned I would talk about this consistency. So we claim our test is a natural test. It's a device that is inserted and being defecated in privacy. But is it kind of like natural feces? So we looked at what is called the Bristol Stool Form Scale, or BSFS. So it's a very simple scale with 7 different types of feces, from hard lumps to the more liquid consistency you would find in diarrhea. So it's really related to form and shape. But a Japanese group led by Matsuda published a paper in 2021 where they measured stool consistency and related it to this form scale. And you would see the data here in the background. So the type 1, the harder one, would have the highest stool consistency, and it would drop more or less linearly on this logarithmic scale with the 7 types. And we wanted to prove we were in the range of normal feces. So we studied the consistency of the device by bending it, by punching on the balloon and so on. And that's what you see in these kind of horizontal diagrams here. And you will see it primarily crosses here at BSFS4, which is type 4, like a smooth soft sausage or snake. So that was really the proof for us that, yeah, we do have a bionics test. This is a natural difficulty test. And I'm just briefly going to show you some data from some of the first devices, so you get an impression on how did it look in the beginning and what did we develop it to. So these are 3 recordings from normal subjects. So A is one person, B is another, and C is a third one. And this is a time plot. So you basically have the pressures, the front pressures, which is closer to the anus, and the rear pressure, which is higher up in rectum. And the blue one is the difference between these two. And this, we asked the subjects to evacuate it here. This subject have like 2 contractions, then it's out in atmospheric pressure. This subject here seems to use 3 contractions. This subject here, there is like 6 contractions. But then we invented a new way to look at these data inspired from cardiology, where the preload, afterload diagrams or the pressure volume diagrams has a huge significance. So we will simply plot the front pressure as function of the rear pressure. So the rear pressure is the driving pressure. This is the resistive pressure. And if you look at the first contraction here, it moves up and down this line, which we can call the line of unity, where the 2 pressures are the same. And I think you can appreciate if you are higher, if the resistive pressure is higher than the driving pressure, there's no way you can evacuate the device. You cannot have a flow going against a pressure gradient. So for sure, you want to move the curve down here. So the first contraction is kind of preparatory, but then you see that there's relaxation here of the anal sphincter, and it comes out with the second contraction. For the other normal subject, see the first one, it works its ways down, further down, and then in the third attempt, it's evacuated. And here you see these 6 cycles. But what is important here is you see anal sphincter relaxation very early on. If you had constipation or dyssynergia, most likely you would not have this kind of relaxation. So this is all normal patterns. And this diagram here, even though it's very simple, is a very useful and simple way to express data compared to these time plots. And here are some examples from patients with constipation. And when I say constipation, these were dyssynergia patients where we knew that there was something wrong with the pelvic floor. So I'm not referring to the patient with slow colonic transit. So it's about half-half. But here you see 4 phenotypes that are distinctly different. This one, very low amplitude contractions, as you can see here, at all time above the line of unity. This is a very dyssynergic pattern that goes in all ways. So at some point, the patient contracts the anal sphincter when he's supposed to relax it and so on. Here, multiple straining contraction, and even though it get way beyond this line, there is still defecation did not take place. So we let the person try 2 minutes, it did not evacuate. And a fourth type here, at first it looks very normal, but then you see the front pressure going up. That's what we call a paradoxical contraction, which is highly abnormal. Again, they cannot control the pelvic floor and relax as we can when we go to the toilet. So those were from the old device. So this is the new graphical user interface of the Spark developed device. So we would have different diagrams. This would be that color topography, but of the diameter plot. So this is where that flip technology come in. Here we have the pressures, here we have the orientation, and this is the bending of the device, so the anorectal angle. And here you would see an integrated view of the device. So here there's one attempt to defecate it. You can see the angle straightens a little bit, and here it slides through the anal canal. And if we look at that in a video clip from a normal person, this is the one contraction. You will be able to see here that it slides through the anal canal. There's a lot of blue field, which means that the diameter is relatively small. And then it comes out, it hangs a little bit in the anal canal. And then it finally kind of falls into the commode. So that was the normal pattern, which we observe in most normal persons. This is a patient with fecal incontinence. So the balloon was filled, but we didn't have time to leave the room. We didn't ask the patient to evacuate yet. It simply drops out by itself at a very, very low pressure. And you may note here, there's no blue color, there's more greenish color, which means that the anal canal has a wider diameter, indicating that the anal sphincters are weak. So enough for what the device can show. So the regulatory process. Our group, we use regulatory consultants. They convinced us this is a class 2 device. It is non-significant risk. It would go the 510K regulatory pathway, meaning that we have to show it was substantially equivalent to an existing legally marketed device. So we selected a device that was already approved on the US market and said, okay, this is our predicate device. And we would test our device against that device. We asked for a so-called Q-Sub meeting with FDA in 2020. And so we basically sent them the material we had, some information about the device, what our intentions were. And they acknowledged this is non-significant risk. It would be the 510K pathway. And they also said, well, you have so much data you have generated abroad. There's no need for animal testing or for any further human safety or efficacy experiments. So they basically took those 200 or 300 human studies we have done and said, okay, this is proof enough. They did suggest a small-scale comparison study with the predicate device. So we would have to do 10 subjects. We did 10 fecal incontinence patients, where we inserted both devices and looked at the data, compared the data. And we also discussed the most important elements with them in the verification and validation process. So what kind of testing of the device would they request as part of the submission? But from 2020 to 2024, there are 4 years. So it actually took 4 years to go through that whole process. And once it was submitted, it was cleared by FDA within 6 months. So in February 2025, this year, we got the clearance. So you may ask, what took so long? And we have to discuss some of the many challenges we met during the way. First of all, there's the FDA-related thing. I mean, FDA are interested in safety and how's the device, is it built correctly according to the specifications you provided? Does it function as intended? So we had to provide hundreds of pages of description of hardware, software, risk analysis, biocom, traceability, electromagnetic compatibility testing, and so on. And we had to set up a quality management system in-house. And of course, we also used consultants to do some of the third-party testing that has to be done by third parties. So a lot of testing is required. but also it has to do with mindset. I mean, I'm not a commercial person. I am by heart a doctor or a scientist, and with an integrated device like this, there are so many possibilities for analyzing data. What 3 tests gave independently before, but in different tests, you could certainly do that with 1 test, where we literally had millions of data that could be analyzed in different ways. More if you think about commercialization, you don't want complicated endpoints. You want very simple endpoints that the average doctor can understand. So, I mean, for example, how long time does it take to evacuate the device, rather than you develop a very complicated measure that gives you some scientific insight. Funding, of course, but also a challenge. I would say NIH Spark has been incredibly kind to us, but funding is a must in the early stages, but there are also delays and limitations. For example, we did not get funding for the second spin. So like the re-engineering of the device that would make the cost much lower. The team, the subcontractors, we are a small team. So if one person would move to another position, it would take months to replace that person. We were understaffed. But maybe the biggest problem that the vendor, the R&D vendor, who would kind of build the device for us or design the device, they were not performing. We had to fire them. We had to take all R&D and production in-house, which took us more than a year to go through that process. That was just bad luck. So we also ran into the electronic component crisis. So for about a year, a lot of the components we used inside the device were not available. Even US manufactured components, some of them we had to find in China, where there were, I mean, some companies had stored some of these devices, of these components. And you also have to consider, if you develop a completely new technology, is that good and bad? Obviously, if you develop a technology that basically does the same as other technologies, it's much easier to pass through regulatory. If you have a disruptive technology, it is more difficult to get it through regulatory. It's also more difficult to get market adoption because people have to analyze or interpret data in a new way. FDA also provided challenges. For example, last year they came up with a new cybersecurity directive. Our device transmit data by Wi-Fi and Bluetooth, and we had to deal with cybersecurity because it's patient data. One of our main mistakes, we kept on trying to perfecting it before we submitted to FDA, but you cannot read the mind of FDA and what they would criticize. So rather than trying to perfect it, make it perfect, then just get to some point and get it submitted. IRB approvals could also be hard. For example, we wanted to study children. About half of the people who comes to me at conferences for our posters and talk, they say you need to use this device for children because there are so many constipated children. But that was really hard to push through IRB. And in the market, doctors does not necessarily want change. They think about how well is this reimbursed? Do you have a PCT code by yourself? And we need to get out and inform the patients. And investments later on is important. Currently, we need $6 million to kind of optimize and go on the market. So for market adoption, what I learned is that patents, prototypes, regulatory approval, they are important milestones, but you don't get acquired because we want to be acquired. We don't want to build a big manufacturing company. So you need to go beyond that. And these potential acquirers in this field, they all say we need to see sales with a positive trend. And as I informed you earlier, that flip technology, it took more than 7 years before, after FDA approval, before they were of interest to the big companies in the market. But it is a good thing to keep them informed. I just been at the DDW meetings. I had meetings with two, where actually all three of the potential acquirers, and we simply share with them what is our goal, where are we, and so on. There's always the question about reimbursement. You don't want to develop a device that costs more than the reimbursement, but can you use the existing CPT codes, or do you need a new code? We believe we can use the existing codes. Distribution is important, competent team. You need to get beyond the validation state, move on to what has value for clinicians. You need to work with early adopters, KOLs in the field, but you have to be aware some of them have conflicts of interest. Identify the countries that accept US FDA, South Korea, Switzerland, do that. And then your device, are you entering a market or are you doing a market expansion? I think our big sales point here is we want to move this closer to the patient because we don't have capital equipment. These other technologies, high resolution manometry, it costs a bunch of money to buy it. And the private practicing gastroenterologists, they do not buy that kind of equipment. They refer them to the major hospitals and they never see the patient again. With this technology, it could be held at the PPD level and they would continue to work with the patient and get the reimbursement too. I think I've been through most of this. We are in an optimization stage. We have set up small scale production, but we don't want to go to big scale production. We set up benchmark clinics with gastroenterologists that we are just about to start. So that's our way into the market, to the market expansion. And I never thought it was a good idea to have one idea, one device, one patent, one company. So you need to diversify your products. So we both have a diagnostic, that's what I've been talking about. We also have a therapeutic version. There was the need for the pediatric probe. Obviously, it has to be smaller. We have developed a prototype and hopefully we have gotten a grant that would support studies. We have a version more for scientific with a video camera on. But a major thing is that device, more or less as it is, could be used for studying pelvic floor pain in women who has pain during sexual intercourse and probably also for biofeedback theory. So in addition to the GI community, there's also an OBGY market. And actually, many of these OBGY doctors also treat fecal incontinence. So I think we need to expand into that market. Plus, very recently, we have developed a swallowable version of the device to study dysphagia in esophagus. Recommendation, I was kind of debating with myself if I should show this or not, because each case is individual. You have to move your way along and kind of decide what is right for me, what is right for this device. You have to go through the same processes. I think the most important thing I learned was that talk with people as many as you can, interact with the FDA, and don't make it too perfect because they would always find one thing or another to criticize. So I'd like to thank you for the attention. Thank you to all the mentors, the support, the teams, collaborators who have participated here. As I said, NIH has been extremely kind to us. The SPARC program, we got a fast track SBIR afterwards. We have our one grants, so it has been good, and we are uploading data. As I mentioned, we have 6 data sets uploaded, 2 models, one on friction force determinations, stress strain properties. So thank you again.